Yogah Karmasu Kaushalam.
Hello, all the participants of the refresher course in life science, which is organized by HRDC Gujarat University Ahmedabad. Myself, Dr. Anjana Kelani, and here I am going to discuss on human microbiome and its clinical significance. Let us go through first the introduction of the microbiome. Microbiome is the term that describes the genome of all the microorganisms, symbiotic and pathogenic, which is living inside or on the vertebrates. Particularly if we are talking about the gut microbiome, it is comprised the collective genome of microbes inhabiting in the gut, including the bacteria, archaea, viruses, and the fungi. It gives the informations about the microbes living in our guts and is growing at the tremendous rate. We are finding the various different types of the microorganisms amongst the human populations, and it harbors the trillions of the bacteria which is present into the human microbiome. From this microbiome, we can get the concept of the core microbiome, that is the set of all microbial taxa or the genomic and the functional attribute associated with the host or any of the environment which is of our interest. Looking to the particular human microbiome, the person's core microbiome is formed in the first 5 years of the birth, and then after gradually it will be changed on the basis of factors which will be affected, including the diet, medications, and the environmental exposures. To study a microbiome, the basic two terminologies can be there, that is microbiota and the microbiome. What is microbiota? Microbiota is the ecological communities of commensal, symbiotic, and pathogenic microorganism, a specific environment. And while the microbiome is what? A collection of genetic material from the microbiota. We all know as a microbiologist or as a life scientist, we should know the importance of the normal flora which is present into our body that can be useful for the synthesis of the various different kinds of the vitamins like vitamin K and vitamin B12. It can be useful for the prevent and colonization of the pathogen from in our body. It may act as a antagonize and other bacteria, the production of the substance which inhibit or kill the known indigenous species. It stimulates the development of the different types of the tissues, and it also stimulates the production of the cross-reactive antibodies. So there will be a different types of the functions of the normal flora in our body. And that's why one can be interested to study these kinds of the normal flora. Now, how we can study this type of this flora, or we can say a microbiota? There are two different kinds of the approach. The very basic approach that is known as the traditional approach, or we can say a culture-based approach. In this culture-based approach, we can collect the samples from the human body. Different types of the samples can be collected. From that samples, one can go for culturing by utilizing the different kinds of the microbiological media. After that, one can go for the identification by the various methods, morphological and the biochemical characterizations can be done. And for these kinds of the classical microbiological identification, for a single colony, one has to do the same thing, or more colonies, they have to do the same things. So it may take a time for the numbers of microorganisms to study which is present into the human body. So, to overcome from this process, one has started to study a metagenomics. With the help of metagenomics, one can study at a time all the microorganisms, and particularly those microorganisms cannot be cultured, cannot be cultivated into the microbial laboratory techniques, or we can say in the microbiological media in a laboratory environment that can also be studied with the help of the metagenomics. Because looking to the data, it suggested that only few thousands microorganisms on the earth has been cultivated up till now. And the data suggests that the more than 30 million microorganisms are existed on the earth, and almost approximately 1.5 million has been described as a species. So this data, like thousands microorganism or the few thousand microorganism, comparatively very less, and yet to study many different microorganisms. And that's why the metagenomic approach will give you the numbers of information which cannot be cultivated into the laboratory. So, these kinds of the microorganisms, how can we study? With the help of the culture independent technique. Here we did not require any kind of the microbiological media. Different kinds of the techniques can be available, but the most widely used technique the people are using nowadays, that is the metagenomic study for the study of culture independent microorganism. So let us discuss in detail regarding the metagenomic first, and then we will come to the microbiome of the human and the human different organs of the human. What is metagenomics? The metagenomics is the study of the uncultured microorganism, because every microorganisms have their own growth characteristics. All cannot grow in the laboratory conditions, and that's why those microorganisms can grow only in their natural habitat, cannot be studied in the laboratory. And to study, to get the informations of these types of the microorganisms, we can go for study by the metagenomic approach. So metagenomic is what? The study of the collection of genetic materials from the mixed community of the organisms. Here we are not having any kind of the pure culture. We are collecting the samples from the environment, either that is the environment or either that is the any kind of the animal. If we are talking about the human, then the different kinds of the human samples are there. So metagenomics enables the study of all the microorganisms, regardless whether they can be cultured or the not. And with the help of the NGS techniques, with the help of the nucleic acid samples, one can study all the different types of the microorganism without culturing into the laboratory. So this is the basic things in the field of the metagenome. Let us have some brief history before starting in detail. In 1970, the concept of sequence comparison come and Needleman and Wunsch algorithm has developed by the scientists. After that, Sanger sequencing has been started, and the day of Margaret Dayoff has started to develop the database. So in 1917, sequencing approach has been started. Earlier it was difficult to get the sequence, but nowadays DNA sequencing is fast and it produces a lot of data that is also required the different types of the computers to save all the data and to save all the analyze the sequence and to compare the sequence. So that's why the database development has been fast in the 1980s. This was the first genome has been completed with the help of the sequencing technology. In 1980s, the development of the protein database and the nucleic acid sequencing database has been started. NCBI, that is nucleic acid, uh, and the EMBL database has been started. In 1990s, the word metagenome was derived by the scientists, and in 2000, the first time microbiome word can be existed. Afterwards, in 2008, and human microbiome project has been started, and onwards 2010 the various different kinds of the platforms are available for the sequencing. And you can see over here that in 2010 Illumina HiSeq sequencing technology platforms was available, and with the help of that one can go and easily sequence that data. Looking to all the technologies, the one word was came that was known as the omics. Now what is omics? Earlier the study was carried out with the help of the traditional methods, and nowadays we are using the different kinds of the molecular techniques to study the same organs, same organisms, same data can be analyzed with the help of the latest technique. So omics word was came from that. And on the basis of different types of the samples which we are analyzing, we are having the different types of the omics technologies. Like genome, we are studying the genome, then it is known as the genomics. Proteomics came from the protein. Transcriptome study, that is known as the transcriptome. Virome study, that is known as the viromics. So different types of the omics studies can be there. We can study with the help of the different types of the molecular approach. It requires the bioinformatics platform to study all the different kinds of the molecular data. And that's why the bioinformatics techniques can be available to study all the omics technologies. Now let us come to the main part, that is how to study a metagenome. The metagenome is the total set of the microbial genome. One can isolate the genome from the mixed sample. There are various different approach of the study. You can have the RNA as a sample, or you can have the DNA as a sample. If it is RNA, then we can say this is the metatranscriptome analysis, or we can say this is metatranscriptomics. If we are interested in directly the DNA extraction, and you can go for the DNA analysis, one can have the metagenomics if you are studying the whole genome. If we are only interested in the particular gene which is considered as a targeted genome sequencing, then it is known as the amplicon analysis. So the basic of the metagenome, or we can say the techniques of the metagenome, can be useful to study the any microbiome. So let us go through now the microbiome analysis or microbiome workflow, how it can be analyzed. Looking to the overall picture, or you can say the big picture, you can collect the microbial samples. You can have the any of the different types of the samples. From that you can collect the samples. You can generate the meta-omics data. You can go and process the data, and you can analyze the data. So this is the overall picture of the microbiome. But when we are looking to the specific type of the samples which we are extracting from that, for example, we are only interested in some of the marker gene, presence of the marker gene. You can go and extract the DNA. You can amplify with the targeted primers. You can filter the error and build the clusters and go and find the diversity analysis. So you can start with your study with the marker genes. One can also start their study with the complete genome. So you can directly have the samples. From that samples, you can extract the DNA, sequence the random fragment, then go for the sequencing process, and at last after the sequencing you will get the small fragments, and you can have the assembly process. By assembly you can have the complete genome samples, and then you can find out the diversity along with the functional analysis. Now in the case of marker genes, you cannot go for the functional analysis, while in the case you can only go for the diversity analysis, while in the case of the metagenome analysis, because here we are studying the whole genome from the samples, you can go for the function analysis of any of the microbials. Instead of the nucleic acid extraction, you can also go for the RNA extraction. Then after you can have the cDNA, you go for the quality checking and go for the analysis of the gene expression as well as the function. So transcriptomics is the different than the marker and the complete genome analysis. One can also study the metaproteomics. The metaproteomics is the different than the metagenomics. Here instead of the genome extraction, one can go and extract the total protein from the cell. Here the protein of the human cell will also come along with the microbial cells. Whatever bacterial cells are present into the samples which can be extracted, from that the total proteome will be going to extract, separate all the proteins with the help of the different proteomics techniques like the 2D gel electrophoresis or the mass spectroscopic analysis. Then after you go for the identification of the protein and analysis of the protein. So if I'm looking for the microbiome analysis, study of the microbiome, any of the microbiome, for example, the human microbiome, I should know that what level of the informations I required, and on the basis of what level of the informations, you can select your samples. You can go for the DNA extraction, whether the RNA extraction or the protein extractions. So here you can select any of the approach for the metagenomics study. If I will select the targeted metagenomics, I can have the two different approach, that is 16S rRNA and the 18S rRNA. If I'm only interested in the microbial diversity like bacterial diversity, I will select the 16S rRNA. But I need, along with the bacteria, I also interested in the other microorganism, whether the fungal cells are present or not, or any other eukaryotic cells are present or not, then I'll also go with the 18S rRNA. So this is regarding the target metagenomics. While other can also go for shotgun metagenomics, which will give you the whole genome data, and along with that one can also go for the functional metagenomics. Let us first discuss on the amplicon metagenome analysis, or it can also be considered as the target metagenome analysis. This amplicon sequencing is also known as the targeted metagenomics, that is a very cheaper and faster process. With the help of using PCR amplicon, one can go for the identification of the microorganism which is present into the sample. What here we need to do is we need to collect the samples, environmental samples or any of the samples. We can go for the first DNA extraction. From the after the DNA extraction, one can go for the PCR amplification with the help of the primer, which can be only amplify the particular gene sequence which is considered as a 16S rRNA gene. So after this 16S rRNA gene amplification, that amplified product can be go for the sequencing process. The sequencing techniques can be available with the different platform. You can go for sequence, and then after you can have the sequence of that particular 16S rRNA. After that, getting the sequence, one can go for the metadata analysis. In that metadata analysis, all the known data are available into the database. You can go and compare the data, your unknown data, with the all available known data into the database. And then after, you can go and identify your unknown microorganism which is present into the sample. So here we can have the idea how that 16S amplicon can be possible. The different types of the regions are there. Particular V regions, variable regions are there. V stands for the variable over here. And primers can be synthesized according to that specific region of the variables. We can select any of the pair, whether it is V3, V4 or V5 regions, and we start the amplification with the help of the PCR reactions. So afterwards, we can get the amplified region, and amplified samples will go for sequencing analysis. This is the whole protocol. Here we can see that the targeted sequence between the V3 and V4 can go for the amplification. Between this V3 and V4 region, almost 459 base pairs are there. The primer which is specific to this region can be useful in the PCR reaction, and then the whole amplification process will be done. At the end of the process, we can get the numbers of amplified product, then go for sequencing, and after sequencing, we have to go for identification of the all unknown fragments. Now this kind of the 16S rRNA sequencing can only give the taxonomic identification. It will not allow to give us the functional identifications. If one is interested in the full genome sequencing, because sometimes we are interested in the functional analysis of the microorganism which is present into the sample. So at that time, instead of the targeted sequencing, we have to go for shotgun metagenome analysis. With the help of the shotgun sequencing, we can go for the genome sequencing. Here, first of all, we need to isolate the complete genome from the samples. Then random fragmentations will be carried out. this kinds of the random fragmentations will be done with the help of either the chemical procedure or enzymatic reaction or the sonication methods. So all the fragments now ready for the library preparation. After that library preparation, it can be go for the sequencing process, and at the end of the sequencing we can have the different fragments of the sequence. Now, how it can be analyzed? It has to go for the assembly process first, and that can be used for the coverage analysis. Here you can see that all the fragments has been analyzed with the help of the similarity sequence similarity method, and it can be organized in a particular way by which you can see that some of the region are the similar in all the sequence. So it can be found with the help of the coverage method, and by that way one can start the assembly process. So at the end of the assembly process, you can have the long read of the sequence of the genome, and lastly we can find our whole genome of the microbial samples. But at the time of the assembly and coverage samples, how we can select the good numbers of the data, or how we can get the idea that this fragments will go for assembly. For that we need to see the coverage. Here you can see that the almost all the nucleotide which is present into the fragments are the similar one. So it can be considered as the 6S coverage, and this will be considered as the 100% similarity. So it can be extended the sequence, and the complete genome sequence from this nucleotide to this nucleotide can be generated with the help of these kinds of the coverage. So this is known as the 6S coverage, and the 100% identity will be there. But whenever you will find this kinds of the analysis that where one nucleotide is not similar but rest of the nucleotides are similar, this can be considered as the 5x coverage, and the percentage identity of that sequences fragment is are the 80 percentage. Then it comes to the 2x coverage. You can see over here that only two nucleotides are similar, rest of the sequences are not even found over here. So that gives the data like only 50% identity will be there. So for the assembly process, one has to find out the 6x coverage so you can have the better result, and at the end of the sequencing process one can have the numbers of read. With the help of assembly you can define the whole genome sequence. So initially we have started our experiment with the natural samples. The samples may be from the environment, maybe from the any of the animals. You can have the samples, you can go for DNA extraction. Here we have targeted only the bacteria. One can start with any of the microorganisms, because in the microbiome we can consider the not only bacteria, it is fungi, it may be any of the eukaryotic microorganism, bacteria, archaea. After that, DNA extraction, sequencing, and the read analysis. So you can have the different numbers of the genome now available with that. And with the help of the different types of the platforms, you can go for sequencing technologies. So earlier only two techniques were available, that was Maxam-Gilbert techniques and the Sanger-Stick techniques. But afterwards technology was developed, and the now next generation sequencing has been came. Then the pyrosequencing, then the reversible termination technology, then sequence by ligation based chemistry, complementary semiconductor based technology, SMRT, as well as the Oxford nanopore sequencing technology is now available for the sequencing data. So here you can see the different types of the sequencing machine has been illustrated, and the year-wise development of the sequencing techniques can be observed over here. So these are the platforms which can be available nowadays. Now how we can select that what kind of the platform is good for us for the sequencing. Certain parameters needs to discuss over here for the sequencing technologies, that how the numbers of reactions can be carried out at a time, what kind of the read length can be developed after the sequencing process, and what numbers of total data output can be carried out over here. Looking to all these characteristics over here, we can see that the Illumina can have the good results. At a time, almost the numbers of 25 lakhs of the number of reactions can be done at a time. The read length of the sequence is the 400 base pairs, and it can be generate almost the 10 GB of the data. So this data GB can suggest that the almost all our DNA samples can be sequenced at a time. Looking to this 400 base pair over here, it will give the 500 base pair. So length of the data, length of the sequence can be good over here. So it can be very good when we are assembling process, when we are go for assembling and when we are deriving the full genome. But the data will be generated over here with this technology, that is nanopore technology, that will be very small data. Over here it gives the lot of data and lot numbers of sequence can be generated. So only the thing is the coverage and the assembly process will be the lengthy task over here, but that can be solved with the help of the softwares, bioinformatics software. So overall technology suggests that Illumina can be the good platform for the sequencing organisms, sequencing nucleic acids. Now at the end of the sequencing process, what kind of the output we will get? We will have the different kinds of the output. The particular data in terms of the ATGC order we're going to have. Now this ATGC order will not give the any of the informations. We cannot get what kind of the genes are present, or what kind of the microorganisms are there, or what kind of the functions this protein will going to done when it is there in the cell. So that this kinds of the data should be analyzed properly, and it could give the assigning of the informations. We can give the assigning of the information at the two different level. One, that is the functional level, and another one is the taxonomy level. Taxonomy level and assigning of the data, assigning of the informations give that what number of or what type of the microorganisms are present in our samples. And the function based analysis that can only done when we go for the shotgun sequencing. Function based sequencing can give you the data or the information that what kind of the metabolites, what kind of the proteins are there, and the respective functions of that microorganisms are what. So two different kinds of the level of information assigning will be there after getting the sequence. Let us see what kind of the tool can be used for this metagenome analysis, or how we can get the microbiome data. The different data analysis tools or the platforms are available. The MGRAST, QIME, or the STAMP, Metagenesis, PAST are the platform where you can upload your sequence data. The sequence data can be available in the FASTA-Q format. All these data analysis tools are available on web-based. You can take a subscription of all the platforms and you can start your data analysis. R is the command based language based analysis tool. With the offline R Studio softwares, one can go and analyze the data. Generally, people are using two most favorite pipelines for the or the platforms for the analysis, that is the RAST, or we can say MGRAST, and the QIME. MGRAST is the metagenomic rapid annotation using the subsystem technology, while QIME is also a kind of open source bioinformatics tool which can be available on the web. You can start your data analysis by uploading the FASTA-Q sequence file. But before that, the FASTA-Q sequence file should pass from the QC, and the QC passed read length sequence can be now start with the analysis. Again, in that analysis, two different kinds of the analysis can be done, that is taxonomical analysis and the functional analysis. If you have done only the targeted sequencing with the help of 16S rRNA sequencing, you can go for taxonomical analysis. If you have done with the shotgun sequencing, you have a full genome sequence data, then you can go for functional analysis also. So MGRAST can provide both the kind of the analysis, taxonomy, as well as the functional analysis. This is the platform of the MGRAST where you can upload your sequence and start your analysis of the data. After analyzing the data, what kind of the output you are getting. Suppose you are working in the human samples, then you can finding the different types of the bacteria are present into the human samples, because we know that the various different types of the normal flora are present in our body. So after getting the data analysis, you can submit your data to the database, that is EBI Metagenomics. You can submit, you can upload the data on your name, and whenever you are required, you can download that data also from this database. Now, why we are doing all these things? Why the metagenome? Why the microbiome analysis? Why the different techniques and tools are used for the study? Because the microbes are present and what they are doing, right? We are interested to study, like in the human body, the numbers of the billions of the microorganisms are present, where they actually present and what the functions they are doing in our body. For that purpose, we are doing the whole microbiome analysis. And to know all the microorganism which is present in our body, the Human Microbiome Project has been started by the US government. After the Human Genome Project, they have started the Human Microbiome Project. The aim of this Human Microbiome Project was what? That what total number of bacteria or the normal flora are present into the human body, on the surface or inside the human body, and what kind of the impact will be there on the human body. So the Human Microbiome Project was initiated, and after getting the Human Microbiome Project, the data has been suggested that the lot many numbers of the microorganisms are present in the human body. According to the different types of the organ, we can see that the different types of the microorganisms are present and they have their own impact into that particular human system. And that's why we are interested to study the different types of the microbiome. Looking to the data, we can see that our mouth contains more than 600 species of the bacteria. The skin also contains the more than 600 the species. If we are taking the samples from the intestine of the colon samples, almost the 8,000 genera will be there. And looking to the vagina, if we collect the samples from the vaginal swabs, more than 200 numbers of the species will be there. So the major numbers of the microorganisms or bacteria are present into the intestine. Almost the 8,000 genera are present, and that's why we are interested to study the gut microbiome or the microorganism present into the intestine. Looking to this human microbiome data, and if you go through the numbers of gene studies from that microbiome, it will be almost more than compared to the human. So this suggests that if we start the human gut microbiome study, compared to the human genome, it contains the lot many numbers of the gene. You can see over here 2 to 3 million genes are there, while over here 25,000 genes are present. So comparatively, the microbial genomes are larger than the human genome, the data suggested. So let us start the discussion on the human gut microbiome. What are the microbial are present into the human gut and what kind of the impact of that microorganisms in our body. After the getting microbiome project, human microbiome project, the data has been generated, and particularly for the gut microbiome, the data suggested that the complete weight of the human, if approximately 50 to 80 kg, then the total microbiota of the gut contains the 2 kilograms. So that may be the good numbers of the microorganism present into our intestine. And that if we consider the one human, then the total number of species, that is 1000 to 5000 number of species are present, and the cell numbers, that will be 10 raised to 30 to 13 to 14 numbers of the cells are present. And looking to the gene numbers, 24,000 approximately genes are present into the human genome, while more than 4 lakhs of the genes are present into the human gut microbiota. And that's why we are interested to study that human microbiota, because this much numbers of the gene may have a good impact on our human health. They may involve somewhere into the our metabolic process, and Looking to this figure, you can see that at the time of the birth, or we can say in the fetus, it can be considered as a completely sterile. There is no microorganism present into our body at the time of the fetus when we are unborn. But at the time of the birth, it start the contact with the outer environment. Very first contact of the outer environment is nothing but the vagina, from where the baby will going to birth. So the vaginal microbial flora of the lady will going to contact with the kid, and that will increase the microbial flora. Then after in the feeding, that will be the breastfeeding or the different kinds of the solid foods will going to provide, that will also increase the numbers of the microbial flora into the baby. Then in the as the age will increase, the flora diversity numbers and the diversity will going to change. You can see over here. And in the case of the adult, we can have the different flora as compared to the early adler age. The flora will going to change. So this will suggest that during the lifespan of any individual, the different numbers and the different types of the microorganisms are present into the gut, and because of this diversity, that may be highly affected to the human health. So these are the phases of the early gut colonization. You can have the four different phases. As we know that the phase 1 is the sterile gut, because before birth there was no bacteria, so the phase 1 can be considered as the sterile gut. The phase 2 is considered as the initial acquisition from the vagina, from the stool, or from the outer environment, like after in suddenly the hospital environment will also affected. So this will be the phase 2, where the kid will come in, the infant will come in contact with this, and it will have the different types of the flora. The phase 3 will be the breastfeeding or the bottle feeding. It may be anything. At the time of the breastfeeding, the particular microorganism considered as the bifidobacteria. This is the important bacteria which will be the normal flora of the human microbiome, and during the breastfeeding, that will be transferred to the infant's gut, and by that way the intestine of the infant will start the colonization of the bifidobacteria. And the phase 4 will be the starting of the solid food, and that will be also considered as the adult flora. Now this bifidobacteria can also be considered as the probiotic bacteria, and we all know the importance of the probiotic bacteria into the intestine, that will be improve the digestive system of the human intestinal or the gastrointestinal tract system. So this will be the important bacteria which will be transferred during the breastfeeding of the baby. So these four phases of the gut colonization is important over here. Now what we do, or what we can get the informations after getting the gut microbiome analysis, how it can be impact on our health. Let us see. Looking to the intestinal microbiota, more than 10 raised to 14 microorganisms are there into GI tract. It may be making us 90% bugs and 10% us. So microbiota, we know that the collective microorganism, and microbiome is the collective gene of the particular microorganisms. It may also possible that amongst all the microbiome or the microbiota, all microorganisms are not good microorganisms. So that can also be go for the abnormal condition creations. So that's why when it increase the different types of the microorganism which can be considered as the abnormal, the dysbiosis will be carried out, and this dysbiosis will leads to the different kinds of the common disease. This kinds of the dysbiosis will leads to the common colon disease or the inflammatory bowel disease. It can also go for the creation of the site specific microbiota into the intestinal. Now looking to the study of gut microbiota with the help of two different approach, we can have the two approach we have earlier discussed, that is functional genomics or the targeted genomics. If you are only interested to study the diversity, presence of diversity, you can go for taxonomy. After getting the taxonomy analysis, you can see that different part of the GI tract have the different numbers and the diversity of the organisms. So, getting this diversity into the gut microbiota, we know that the numbers of different types and the diversity samples are there. And you can see that the common of all the microorganism present into the gut is the lactobacillus, right? Other organisms are also present, but the lactobacillus is commonly found at complete intestinal tract because this lactobacillus can produce the lactic acid, and that acid formation can be useful for the intestinal tract. Looking to this gut microbiota during the human lifespan, we know that as the ages is increased during the age, different age, we can have the different numbers of the microorganism present into the intestine. The analysis of the different regions can also be suggested that the different diversities. Over here you can see in the figure that the different three regions has been collected the samples from the Europeans, Americans and the Asian human population, and the gut microbiome analysis has been done, and the data has been suggested that the population and the abundance, means the numbers of that species, is been varied into the different. So region wise also we can have the different types of the diversity into the intestinal tract. Now the gut microbiome appears altered into the many chronic diseases. We can see over here the different organs of the our body have the different disease. Uh, along in this 10 causes, almost the 7 cause leads to the death, and the data suggested that all the different kinds of the cause will be affected by the numbers of microorganism, or we can say the gut microbiota is somewhere correlated or somewhere influenced by this analysis. You can see over here we have started the analysis of the sample collection from the healthy and the patient. In the case of the healthy samples and the patient samples, you can see that in patient samples we can find the numbers of diversity of the unnecessary or the different types of the pathogenic microorganism, while in the healthy human being or the healthy gut it cannot be present. So the common alteration in the loss of microbial richness. This can also be studied that in case of the severe disease of the liver cirrhosis, you can see this one of the two different kinds of the sample has been started to study. In one, the patient healthy liver samples has been collected, and in another one is collected from the liver cirrhosis disease. In both the cases, the diversity of the microorganism will be the different. Along with that, liver cirrhosis, the oral microbiota, not only the intestinal microbiota, the oral microbiota has also been studied, and that also suggested the diversity in the patient as well as into the healthy human being. Now, this will leads to the decrease into the good bacteria. The microbes poor into the gut, microbiome less healthy. We can see that if the pathogenic microorganism will start to grow in a high in number, that will going to suppress the our normal flora, and that's why the lactobacillus kinds of the microorganisms will be decreased in the numbers, and that will leads to the low butyrate production, high lps production, high h2s production, and oxidative stress increasement into our body. Because this microbiome study of the gut is correlated with the different types of the disease of the human, we should know that microbiome of the gut is the important one, and microbiome richness is associated with the health and the well-being. So it is better to be rich than the poor, and that's why we can start the improvement of the gut microbiota. Integration of the clinical phenotypes, microbiome, and metabolome data reveals the microbial species importance for a disease. And as they are correlated into the different diseases, this kinds of the microbiome restoration can be done. This microbiome restoration can be done at the two different level. One can go at the molecule level. You can have the good types of the probiotic, or you can start the consumption of the amount of the fibers, or you can go with the restoration directly with the microbes. We can take a different kinds of the product which will provides the good bacteria in terms of probiotic products. You can take and you can restore the microbes. Nowadays the new concept has been arrived that is known as the concept of poop pills. Now this poop pills is nothing but the capsules which contains the stool samples from the healthy human being, which contains the good numbers of the gut microbiota, right? So from that kind of the poops, the samples collection and the freeze dried capsules has been prepared, and this kinds of the capsule consumption can increase the gut microbiota. So the concept of poop pills is nowadays carried out, and in the US one of the pediatric institution has started the treatment of the pupils into the initial treatment, the kids who is suffering from the diarrhea, and they have got the 90% of the success to control the diarrhea, because diarrhea we know that the loss of dehydration, and that is completely related to the intestinal tract disease. By restoration, by the improvement of the gut microbiota, one can solve the problems of the different types of the disease. One of the disease cured over here, that is diarrhea. That has been the approach has been started into the USA. One of the another approach is the fecal transplantation therapy. From the healthy person who is having the good microbiota, we can collect the stool samples of that particular person. You can go for the purification of the samples, and after that you can have the particular consortium of the microbiota which is collected from the stool, and that kind of the consortia along with that particular fecal samples, either you can prepare the capsule or you can provide to as a direct therapy to the disease caused person. So the fecal transplantation therapy is also one of the latest therapy to improve the microbiota, gut microbiota. So this US country has started this therapy, but it is in our tradition. I'll give you one of the concept over here, the Panchgavya. Panchgavya is nothing but the five different components of the Indian cow, right? So Panchgavya is the oldest technique by mixing the five different kinds of the components of the cow. The product can be formed, and that product can be given to the patient for improvement of the microbiota. One of the component of the five components, milk, curd, ghee, along with that, one of the component is the cow dung. As we have the numbers of microbiota in our intestine, the cow also have their own microbiota that will comes to in the cow dung. By providing this Panchagavya treatment, all the microbiota of the animal intestine that will transfer to the human being. So our Indian tradition earlier also following, now US has started this technology. So I must suggest that this kinds of the Ayurvedic techniques we need to follow for the development restoration of the microbiota. In our laboratory, we have started to study this Panchagavya metagenome analysis, and we have found the many number of the microorganisms which can be useful for the development of the gut microbiota. So data analysis of the Panchagavya suggested that number of microorganism, out of this microorganism, bacterial bacteria the many bacteria can be useful for the improvement, and that can be directly associated with the human health. So this is regarding the gut microbiome. After the gut microbiome, another important microbiome in our human body is vaginal microbiome. Let us discuss on this human vaginal microbiome. So we know that the microbiome in the reproductive system is the very important part, as we know that the different organs of the human contains the different types of the normal flora. Along with that, vagina also contains the different floras. So the genital tract of the female body, like vagina, contains the different types of the organisms. And we can how we can start the study of these kinds of the microbial flora. You can collect the samples, swab samples from the vagina, and we can start the DNA extraction, nucleic acid extraction. Again, you can have the two approach. Either you can go for the targeted analysis if you are only interested that what type of microorganisms, what type of bacteria are present. You can only go for targeted analysis. You can go for sequencing. After sequencing, one can go for data analysis, and you can find that what type of the microorganisms are present. You can go for both the type of the targeted analysis, 16S analysis and the 18S analysis, for the identification of the microorganism present into the vagina. So after getting the analysis over here, you can see that the graph suggests that this blue color is the gut microbiota, red one is the oral microbiota, and this yellow color suggests the vaginal microbiota. So this will also have the idea that the numbers of microorganism or the various numbers of species are highly in the presence of vagina. This graphical representation is also suggests that the particular type of microorganism only found in the vaginal samples, not into the another samples like the gut samples or the mouth samples or the skin samples. So these are the microorganisms like lactobacillus or the bifidobacteria or the another different types of the microorganism which is only habitat into the vagina, not into the another human organs. So this microorganism may have a specific impact on the human vagina. Let us discuss in detail regarding that. Whenever you collect the samples from the different region of the reproductive organs, reproductive system, the diversity may change. This data suggests that if you go and collect the samples from this region, you will only find the majority of the lactobacillus microorganisms. While you go and collect the samples from the other part of the reproductive tract system, the diversity will be increased, the high diversity will be there. So distribution of the vaginal microbiome into the different place, different location into the tract will be the vary. And this type of the microorganisms have their own different types of the impact. Looking to the vaginal microbiota, there are different 5 kinds of the groups are available, and this 5 groups can be distributed into the different location into the tract. This is the how we have started in our laboratory, the sample analysis and all the things. We have done the 2 different kinds of the genome extraction. We have collected the samples of the 2 different human vaginal sample, one of the healthy vaginal and one of the infertile vaginal samples. And then after from that samples, we have started to extract the DNA. Then targeted sequencing has been done. Targeted sequencing in 16S rRNA V3 V4 region has been amplified, and then amplified product will go for sequencing analysis. After sequencing analysis, you can go for the analysis. This analysis suggests that the various numbers and the abundance, various diversity will be found into the vagina also. This, this will be the bacterial diversity found into the vaginal samples of the healthy human. healthy vagina. In that healthy vagina, we have started the different level of the analysis, and you can see that the Lactobacillus family organisms are also found in the high numbers. So that will suggest that the Lactobacillus is also part of the human vagina. According to the gut microbiome, it is also suggested the gut microbiota is highly abundant with the Lactobacillus species, and vaginal sample is also contains the various different species of the Lactobacillus genus. So somehow they are connected with each other. So let us see what are the clinical significance, why we are interested to study the vaginal microbiome. What is the clinical significance of the vaginal microbiome? The human vaginal microbiota contains the Lactobacillus species, and in the different age woman, the numbers of and the species, different species are vary. We have studied that in the case of the adult, in the case of the menopause, before menopause and the after menopause, the different numbers and the different types of the cells. So that suggests the ages also matter in the case of the biodiversity of the vaginal microbiota. Along with that, this type of the species of the Lactobacillus cryptosus associated with the lower risk into the HIV infections. So we can say that this type of the Lactobacillus species or any other good bacteria which is present into the vagina that will restrict the entry of the microorganisms like the pathogens. So that can be helpful. This is the data analysis of the healthier pregnancy and the infertile pregnancy. You can clearly see over here that the diversity in both the patient in the healthier and the infertile patient is the different. The vaginal epithelium of the reproductive age woman is contains the high in the glycogen, and this high glycogen provides the different source for the feed in present bacteria which is present into the vagina. So as high in the glycogen present, it can be good for the present bacteria into the vagina. Along with that, estrogen can also boost the lactobacillus colonization. So this is completely the hormone production. So this hormone production can boost up the lactobacillus organisms. So at the different age and at the different level of the different phase of the life, like before after menopause, the estrogen level production will be different, and that's why at the time of that phase of the life, the presence of the bacteria will be vary, the data analysis suggests. And the overall role of the vaginal lactobacilli is what? This epithelial lineages will promote the growth of the lactobacillus cells, and this lactobacillus cell when produced in a high amount, that will affect the pH of the vagina because the production of the lactic acid. This lactic acid produced by the Lactococcus lactis, which is present into the vagina, will decrease, which makes the acidic environment over here. And in that acidic environments, other pathogen cannot grow. So whatever microorganism which is coming from the outside, right, that cannot be grow. And that by that way, the complete reproductive system can be protected by the pathogens. So these are the numbers of microorganisms present into the different locations of the reproductive systems of the female vagina. Why this is important to study the human vagina? Because the one of the case study, the three different kinds of the cases we are having over here, the birth of the baby by the different three techniques. So here we can have the three different cases. One that is vaginally born and the breastfeed baby. Another one is the vaginally born but not breastfeed, and another one is the C-section baby. Now, in all the three cases, we can see that whatever vaginal microbial flora has been exposed to this two kid, but not this two, not this one. So the microbial flora, the normal flora present into this baby may be vary from this two. Again, by comparing these two cases, one is breastfeeded, another one is not breastfeeded. So the normal flora of that particular organ from the mother will transfer to this one, not this one. So by looking this, we can say that the richness of the microbial flora in this particularly vaginally born and the breastfeeded baby is the high. Diversity will be high compared to this. So this is one of the analysis which can be done with the help of the microbiota of the infant along with the vagina. This is one of the case. Another case is the vaginal microbiota of the pregnant woman as compared with the non-pregnant woman and the sexually worker woman. So here also you can see that all three comparative analysis shows that the diversity in these two are also vary. Here, this is non-pregnant woman. The hormone level production is completely different compared to this pregnant woman, and that's why the normal flora diversity and the numbers are different compared to this. And this will be the different uh, case, that is sexually worker person, that exposed by the different different types of the environment, and that's why the diversity has been found highly compared to this two over here. So this kinds of the analysis suggests that the age, the clinical conditions will be most better uh, influenced with this microbiota, and that's why the microbiome, instead of studying the single cell, single species, we are interested in the complete genome analysis, because the single species can play an important role, but many are likely to contribute, right? So this microbiota contribute at the different level. This kinds of the human microbiome research, human microbiome analysis can be have a good impact on the research as well as on the health in the field of diagnostic, in the field of prognosis, patient monitoring, or the target for modulating or improvement of the health. We can get the base of the microbiota informations to better preserve health and better treat the disease with the help of all the informations of the microbiota. And here have some landmark of the human microbiome. So this is all about the microbiome analysis. Here we have discussed the two major microbiome, that is gut microbiome and the vaginal microbiome. Somehow they are also correlated to that, because at the time of the birth, the baby has started to expose the numbers of microorganisms from the mother. So afterwards, that will be enriched into the gut microbiota. So along with that, certain clinical conditions will be directly affected by the microbial diversity present into the human body. That's why it is highly important to study the microbiome. And these are some of the data suggested that lineages, how the development of this field has been started. And last but not least, that we have studied that the microbiome has the good impact on the human health. But not only the human health, it can also have the impact on the environment. It can also impact on the environment, animals also. So by this studying the human microbiome project, which is initiated by the NIH, National Institute of the Health, which is situated in the USA, the One Health concept has been completed. So this is all about the human microbiome analysis and the human microbiome. I hope you have got the good stuff regarding this field. Thank you very much for listening me, and I would like to thank over here the organizing committee of refresher course UGC HRDC Gujarat University. Thank you one and all.